So good morning to most of you, I guess. People in the east of Quebec and Ontario, maybe the afternoon started for you. So yeah, as Maria said, I work at the Ottawa Research and Development Center in Ottawa, and part of Canada Centers. And as Maria said, and Maria's been to Ottawa, and she's been to the center here. You can notice this Ottawa Research and Development Center, the map, the first image you see, and that's the main building we work at. Behind there's all the land that you see here, and people are not were not part of the AFC group, wondering what all this land is for. Typically, breeders here at the Ottawa Center, as well as from other centers, this is where they plant all their lines related to crops that we work with, in, especially in Ontario, like wheat, corn, barley, soybean, of course. So this is all like experimental plots every year. This is probably from I don't know which year it was taken. It's beginning to be like starting looking like this. Just started the cleaning up the land yet. It hasn't been planted yet, so it's still dark. So this is what it looks like in Ottawa. So we're not far away from the city center. So anyway, so that's so just topic in hand. As Maria mentioned, my focus being in Ag Canada and related to crop species, we work on the fungal called Graminearum, Fusarium graminearum, against wheat. As you know, wheat is a major crop in Canada. Of course, disease related to wheat is that caused by this fungal pathogen, Fusarium graminearum. It causes what they call the head blight that you see here. That's a bleaching of the wheat heads. And on the right side is the Fusarium. We study both sides. We study both the what are the factors, the virulence factors that contribute to for the pathogenicity, as well as what are the mitigation strategies that plants use? What are the immune system that plants use to counteract the Fusarium infection? So we're studying both the intercellular signaling pathways in both Fusarium and wheat, as well as the interaction. What happens at surface level? What happens that just when they initiate the interaction, initiate the infection process? What happens at molecular level? That's pretty much my focus of the area in my lab. And as you notice, what the Fusarium head blight, in addition to the bleaching of the heads, also causes all the so-called the shrunken kernels. We said it also, this particular graminear species can also infect corn. You get the ear blight here, and you also infects barley, and also causes what they call a gushing of the. If there's enough toxin made, it causes problems in the barley industry. So the tolerance for that toxin is very low as well. I mentioned in the red here is a disease complex. Even though I mentioned the graminearum is a main pathogen, is involved in Fusarium head blight. But what we know now is that when you harvest infected seedlings like this, what you will see is that many other Fusarium species are in there as well. So either they're somehow contributing to that, to the initial, not necessarily initial infection process, most likely to the disease onset. So some of them are helpers. Some of them are they call piggybacking on. They're pretty much like freeloaders. The whole concept, that's our emphasis, that whole concept, disease complex, is becoming more prevalent in the study of FHB and other diseases for that matter. I think it's important to know that it's not a single entity like graminearum that is involved in the entire disease process. So on that note, I mean, so the question then is, Fusarium species, YDB Fusarium species is huge. It's over 300 species in the, as generalities spread out. As you get the map here, is pretty much around the world and affecting many, many different crops. The graminearum I'm talking about is part of this sambucinum complex. I'll come back to that later. Just as a background, you know, the graminearum species themselves, they can get widespread of diseases, including legumes, cucurbits, some of the ear blight I mentioned to you about, rice, banana, oxysporum, the part of the oxysporum complex also causes the infection on soybean and tomato. So you can imagine, this Fusarium is a very diverse species, and the interesting concept is how does it do it, right? But what are the commonalities between them? Is how do we go and analyze them? That's a bigger question we ask in overall. So more emphasis on more interesting thing is the sambucinum complex itself, where the graminearum is, which we work with, is itself is species rich. It's got a lot of virulent pathogens. I think Iman Laraba had a nice paper a few years ago saying that more than 370 species are involved in part of the sambucinum complex themselves. They impact many cereal crops, as I mentioned, including wheat, barley, maize. And if you look at the genome itself, you'll see it encodes a lot of protein effectors, virulence factors over 600 plus per genome, typically per isolate. In addition to that, they also code for a lot of secondary metabolite. What are they? So question is that, in addition to the virulence protein effectors that cause involved in disease, we already also know that many of the secondary metabolites, small molecules, probably in congruence, in combination with the protein effectors, also contribute disease. In addition to that, in addition to causing disease, they also secondary metabolites are involved in things like the toxigenic potential of these toxins. You see that in your top here. The DON, for example, it's a mycotoxin that is, again, it's a virulence factor because if you knock them out, you get no disease. It also involved in feeder fusion in pigs. What happens is that the toxin induces vomiting of the pigs. Therefore, the pig does not gain any weight, so he loses weight. So farmers lose a lot of money. Another example of a toxin as a virulence factor or a secondary metabolite as a virulence factor is a good example of fusotoxin here. You notice that it is a small thing. If you knock without the fusotoxin, what happens? It prevents Fusarium from going to cell to cell to cell. So the green thing here, that's a GFP labeled Fusarium, is primitive, limited to one cell. As soon as you add fusotoxin, they can start spreading from cell to cell to cell. So it seems to be, so while the DON is perhaps a protein inhibitor, it seems like the fusotoxin contributes to cell-to-cell migration of Fusarium inside the plant cell. So you have a combination of effectors, protein effectors. You have a combination of secondary metabolites. Overall, contribute disease process. So the question we're asking is that by all this species-rich Fusarium sambucinum complex, are there any commonalities? Are there anything associated? By looking at a single isolate, you may not find them, right? You can do some bioinformatics to find out what they are. But ideal to get a bigger scope of how this effectors contribute to the disease process, we said, okay, let's study we initiated the pan-effectome study quite a few years ago, maybe like 5-6 years ago. What we did was what is the point of that? What is the point of the Fusarium sambucinum species compound pan-effectome? I mean, we related to the sambucinum complex. You can pretty much initiate the study any of the other clades that I showed you that this Fusarium is part of, like the oxysporum complex and any other complex as well. So what's the point of this doing the pan-effectome? You will capture the genetic diversity across the whole population. What it'll reveal is that are there differences between the effectors between the isolates, and then are there commonalities? As seen in the Venn diagram here, there's a great example of, are there core proteins that are involved in a generic virulence factor as opposed to accessory that are common to some of them, as well as they're unique to one isolate or the other. So there's typically core effectors may be most likely involved in and essentially a basic infection process. That's one of the hypotheses. And another thing, you think accessory effectors are probably most likely involved in adapting to the host or the environment, and probably a combination of both, obviously. So I think what we focus our effector things like can we focus on the core effectors as an example? For this presentation, I'm going to focus on the core effectors. How do we get at that as we look to the pan-effectome? Are there common effectors among all the Fusarium species that could be used for, come down later on, how that could be used for either surveillance purposes or, for that matter, for mitigation strategies as well. So what is a pan-effectome? How do we do this? So this is pretty much an outline of, and that could be a generic outline. We modeled this 3 years ago, mostly done by Chris Blackman in my lab. All the things I'm going to talk about today, I would say 90% done by Chris Blackman, PhD student in my lab. So we started with, as a starting point, 195 genomes in the sambucinum complex. They included many graminarian species. If you notice the last slide, the sambucinum complex slide, the clade, there are a lot of graminarium in that clade. So we used quite a few Fusarium species and some other species in that clade as well. And then after that, we did gene prediction. You can notice that some have more genes than the other. POI is actually interesting. The graminarium as a genome level, graminarium is very stable. It's got a 4 chromosome, doesn't change much, as opposed to other ACM or POI, for example. Depending on the isolate, it could have a range from 4 or more. depending on isolate. So in addition to, it can have extra chromosomes. They call extra chromosomes or accessory chromosome. Other fission species have. Grammar does not have that. Has on a four chromosome, very stable, pretty much. I would say in terms of model, very easy to work with. So then we used we identified all the genomes, sequence of genomes. You notice that important thing to point out is that not all genomes are complete. This is done 5 years ago. Even now, most of the genomes are still incomplete. The strain that we work with, isolate that we work with, is completely sequenced. It's pretty much we got a PacBio from end to end, like with no ends, really complete genome. There aren't too many like that. There are a few of them getting more and more in the pipeline. When we started this project, we were able to get about, you can see the between 90 and 98% completeness. So not that incomplete. It's very important that I come back to later why that's important. Then, of course, you use the proteome for different softwares. And then secretome, again, we use various softwares. Important thing is that when you use, again, another software called Effector P, is getting it identified how many effectors we identified. So candidate effectors, we identified all the entire complex, over 125,000 effectors. That's a lot of stuff to deal with. So question, what do we do about it, right? So we notice that even give an idea here, even though it's a medium of 645 effectors per genome, for the strain that we work with has about 650 to 800 effectors, but you notice that Cerealis has like over 800 effectors. So that could be due to, again, like even bringing it due to the accessory chromosome that goes from change from isolate to isolate. That could contribute. So we know, for example, POE accessory chromosome harbors a lot of the effector proteins in there, so it can go in and out. So that changes the thing. The number of effectors change from isolate to isolate. That's an important point to make here. So what do we do here? So once we have the 125,000 effectors, how do we study these things? And it's not simple to say, let's study all 125,000. So what you did was get the we call protein ortho families. Initially, we call rare fraction analysis. I'll show you the next slide. I'll show you how that works. Once that's done, we can do the annotation of the limited core effector we can study. There you go. So what is a rare fraction analysis? Essentially, what it is that gives you total effector diversity or pangenome across many, many different strains. If you notice that thing here, what you see on the x-axis, all the number of genomes, 0 to 195 genomes. When you start, let's say we start a genome sequence, we do the count the number of effectors, let's say 645, you count them up there. As we start adding the genome, we started counting all the effectors we added to the genome, added to the effectors as it goes up and up and up. You notice that in the total, as we go to 150, 50 starts leveling off. So what this tells us, two things. As you add more genomes, we can add more effectors. And then more than that, after 160, especially Fusarium species, we don't want to get extra. There's no benefit to sequencing more genome to find more effectors after 150, 160. Bacteria, depending on the size of genome, that's the most critical point I want to make. Depending on the size genome, this rare fraction will change, of course. The bacteria is smaller, and maybe other fungal pathogens are more different. So this has to be done with there are, again, pathogen dependent or fungal dependent thing. So important thing is that they can reach a saturation point, according to this here, right? So what do we find? In summary, overall, we found in terms of pan-effectome, 444 ortholog groups. What mean that is that essentially we took 125,000 effectors. We, based on blast sequence, we said, okay, this belongs to this particular group. Okay, stringent analysis to how many groups they can belong to. And in terms of core effectors, that means what we mean by this is that there are only 50 effectors, 50 families that are found in all the 195 genomes. But because I remember I said all the genomes are not finished, we took a more conservative approach and said, okay, let's say it's 100%, let's say 95%, we came to a number called 235 effectors. So there are 235 core effectors that are common to 95% of the isolates that we analyze. So that's huge. That's 235 core effectors. That's the Venn diagram that you saw before. Then what do we do with that next? So now we're coming from, pan-effectome to 125 effectors into ortho groups, now finally down to 235 core effectors. Even there, if you then you can parse that even more. Here is an example of how the function diversity of the core effectors change. If you notice on the right-hand side of the group, that first of all, the 235 factors that we looked at, 290 are expressed in plant. So we know they're most likely potentially as a function, but we know they're expressed in plants. Are that if you notice that 88 families has zero diversification, that means there's no change in nucleotide at all in terms of percent changes. That means their core effect, they're not changed. They're probably likely very important. Any change, maybe it's not just it may not in one, its role might be in addition to causing infection process, they may also be maybe important function for the fungal itself. For example, if you have enzymes that's important for digesting cell wall, right? Cell wall degrading enzymes, CWAE, you can't do without it, right? So that means you don't want to change the enzyme activity of that too much. So you want to keep that preserved and conserved. We notice that I made the dotted line here, the 44 families that are actually undergoing more than 1% selection, diversifying selection. They're changing, not rapidly, but they're changing 1% or more. So we said, okay, what is that implication of that is? So implication of that, if they're changing at 1% or more, that means they're trying to avoid, they want to keep up with any kind of interaction that happens with the plant, the host, right? So they want to evade the plant immune system. The reason you want to evade it because, again, the plant immune system on the right-hand side, he said there's a redundancy plant immune system. If you go, if the effector comes here, guess what? It's another way to go. So the output could be dependent on multiple different pathways can take for the plant immune system. It's robust, right? That's important to think of. It's not like one single pathway limiting immune response. Many different ways you can get immune. You may not get 100%, but you get enough. That's the thing, right? We always think about immune system be all or none. We got to think of, even a partial resistance may be sufficient for the plant to survive and move on, and enough to pathogens curtail its ingress into the plant. So these are level of interactions important. Like could be quantitative, not like all or none. It be quantitative as well. That's the FHB resistance, all about quantitative resistance. In between all the range of resistance you'll get. So what are the functions? What are the core effectors do? In addition, I mentioned they avoid the immune system. They can also target the hubs in the host networks. The hubs could be here. That might not be hubs in terms of single protein. It could be a protein complex. You heard of resistosome, for example. There's a complex of maybe 50, 60 proteins. So, for example, if you have this was functional redundancy comes from the effector side. Let's say in the protein complex, in the hub here, one of the plant proteins is able to detect one of the effectors. We have the option of multiple effectors, unrelated effector targeting the same protein has impact because you can have a redundancy in the so redundancy in the effector is hugely important because once it's detected by the plant, you'll screw up if the effector function is gone. However, if you have other effector targeting the same protein with a structurally different immune system is bypassed, right? So that's the whole point of the redundancy built in the effector function. And the last thing I want to mention in terms of diversity of core effectors, we talk about, always in the context of effectors, in the context of plant immune system. But remember that many of the fungal effectors, be it secondary metabolized or in protein, they evolved long before the plant came on Earth. That means most of his functions is intermicrobial competition, either to avoid or fight against create a niche for itself, so that against other microbes. Or secondly, we also think the new concept of immunomodulize. Myself and David Overy in our department, what we work on is that there are secondary metabolites produced by the fungi, some other endophytes produced by the fungi. They actually enhance plant immunity. So what that does is, by doing that, it can create an environment where that any invading microbes come in and gets blocked off. Create a niche for yourself as opposed to and block any invading pathogen, thwart the function of the invading pathogen. So the combination of either actively killing the microbes by using molecular competition or in helping the plant to boost the immune system can also way to create a niche for the microbes. So that's the thing. You got to think of how the functional diversity works. So how do we annotate this? So once I mentioned, now we have the core effectors, 44 of them. How do we find the function of these things? So what we did was, one is structural base. So we Tian Lee in my lab group, he did all the structural analysis of this proteins using AlphaFold. You probably are mostly aware of that now. You can do a lot of you switched from primary sequences structural three-dimensional structure to literally find the function. So, for example, here if we give you an idea, the many of the if you look at if you do a normal blast of the 44 effectors from the 44 family effectors, most of them are hypothetical proteins. However, if you do the structural base, if I find new function, for example, here again, example 4 of them here, they actually have function to other known virulence factors. So it's at the primary amino acid sequence, we saw no function, no homology to any protein. However, the structure level is pretty much structurally identical to many known virulence factors. So there's another base called SUS, called sequence unrelated but structure similar. There are about 20,000+ the structure out there of virulence factors that are all in the database that we can use. And so find a structure for your own protein and then do a full seq to find out if there's any structural homology to the SUS database. So you're able to find unknown function of your protein to a known virulence factor in the SUS database. Very powerful technique and saves you a lot of time in the end. Before you couldn't do that. Literally, you couldn't do this like 2-3 years ago, even less than that. So it's a really powerful technique. And secondly, we can use a secretion assay. Remember, the one important function of the effector, they usually secrete out. They actually have to physically get out from the pathogen into the host and screw up the immune system. That's one of the thing. It could go a different way. It can come out directly, or can use it independently through thinking like a vesicle base. So what we assessed here was a secretion assay using yeast-based invertase assays. Very simple assay. We hook up your effector to a protein called SUT2, which is invertase. What invertase does is converts the reducing sugar into like sucrose, for example, into sorry, I can't see that here again. To detect reducing sugar. So that's a non-reducing reducing sugar. So sucrose, for example, into glucose and fructose. It changes color. So if you have the color change, for example, from blue to copperish. So what you notice on the left here, they're all the 44 effectors you see here. Okay, that's all in the 96-well plate. You can see majority, the one in the pink here, majority and all secreted through the normal assay. It actually physically comes up. Some you see it didn't show up in this assay. That is telling us that it could come different way, usually through vesicle base. In fact, we already know that we already looked at some of the effectors that are here that shows to be negative in this assay. We can actually show that it's been shown previously by other people. It comes through as a package in a vesicle, and that gets pumped into the host cell. So this is at least 2 different ways you can make to do the functional annotate your effectors of interest. The third one, we actually more functional characterization. We developed the wheat coleoptile assay, or wheat overlap, as we mentioned. Typically, we take a wheat coleoptiles. You notice that here, there's a wheat leaf here. We clone our effectors independently into the Ustiala hordi, which is one of the smut fungi that can clone the effect. It's a very, very good non-pathogen for wheat, able to inject or able to secrete or able to what they call deliver your effector of interest into wheat independently. So a single effector can be delivered into the wheat. So what we do is we do that. The first injection is that we deliver your effector of interest. We wait for 2 days, like 48 hours. And subsequently, we inject the same area with the DC3000, which is a bacterial pathogen, which we know is virulent on wheat. It actually becomes necrotic. So you can see that here in the bottom here. That's it becomes a DC1000, become bleaches. But if the effector has a function, let's say if it turns on immune response in the plant, that means you get resistance. What you will see is a reduction in the bacterial growth. You see that already in the physics. You can see visually, this is bleached here, the DC1000 on its own. However, in the combination of DC1000 and the effector, what you'll see is that reduction in the bleaching. If you do a coliform counts, you'll see a reduction in bacterial growth as well. So we can quantitatively measure the impact of the effector that's functioning in the host. That's the main thing of overlap assay. It's a very cool way of doing it. So we took all the 44 effectors, and then we did this is a summary. I'm not going to go through in detail, obviously. This is done on such a variety of wheat called Roblin. You notice that the important information is that independently, majority of the effectors, if you look at that, it's on the left side. That means it's able to turn on the immune system, immune pathway in the wheat. And that means reducing the growth of the bacteria. On the other hand, you'll see two of them here are actually able to do the opposite, able to somehow, maybe target a susceptibility factor for that matter and induces, uh, more bacterial growth, becomes more susceptible. And here's a control, SN-tox3, which we know is resistant in Roblin, that can show that it's as a control of this empty vector as well as a usolog vector control. Compared to the controls, you see that many individual effectors can turn on the immune pathway to provide resistance. So something very novel that even though we heard of Fusarium head blight as a disease, as an organism that causes disease, but independent individual effectors on its own, if you overexpress them and send it to wheat, it can actually turn on the immune response. Very interesting. So we also did that same thing in a resistant variety called tenacious. I won't go into the detail, but also the next slide is that it's a summary of comparing both the tenacious, which is a resistant variety of wheat, versus susceptible variety of Roblin. That's in pink in the Roblin, and the one in the blue, or the, I guess, teal as in tenacious. What you see is two things. Once you see a quality of resistance, for example, some effectors, okay, like O2386, for example, causes disease, in Roblin, but has no impact on tenacious. Okay, so it's one-sided. We also see qualitative. Some effort like 4848, for example, causes susceptible in Roblin, but more resistant in tenacious. So very opposite. So you have a qualitative resistance depending on the cultivar. But you also have this quantitative resistance. That means some effectors can induce resistance in both the cultivars. So you have, if you notice at all the the colony forming units are on the left side of the hashed bar here, telling you that both are all these effectors, when delivered, cause resistance in both the cultivars. So you have a combination of both individual. It's a bottom line, as Reiko mentions, that when you express individual effectors in different cultivars, you get a combination. You get both ETS, which is effector triggered susceptibility, which is that susceptible, or you can also turn ETI, which is effector turn immunity, that means induces different response in plants. Okay, so that's something really exciting we discovered. So the question is, remember, this is done individually. That means the effectors express individually through the Usalago system. What happens in the context of the entire organism? As I mentioned to you, remember, Fusarium harbors over 650 effectors. What happens do these individual effectors have any impact on in the context of the organism? Here's an example of that. So here you have, this is transgenic Fusarium. That means these genes, the family A, for example, or all these family members, they've been knocked out. Individual effector has been knocked out in the Fusarium itself. The transgenic Fusarium with the deletion of this individual effectors. What you see is interesting. Here's a family A that causes, when you knock them out, it increases the disease. Interesting thing is, the same guy does not do anything in the tenacious. However, it also is your disease in durum wheat. So you have this single effector, again, differential effect on different cultivars. You also have family 29. Again, seems to be more disease in tenacious, no impact on Roblin. However, impacts on disease in tenacious. That tells you we're targeting a combination. Depending on the variety of host, you're either missing or a presence of susceptibility factors that may be contributing the phenotype you're observing here. So even individual effectors have an impact. Core effectors have an impact on disease outcome. Okay, that's something we never thought of before. This is something new that we've been discovering, how it can the effectors have single effector, even in the context of Fusarium organism, have an impact on resistance susceptibility in the end result. So in the end, what we have in the Panopticon summary is that we have 44 effectors, the core effector families, that are undergoing diversified selection. That means 1% above, they're changing, i.e., they're recognizing something the host recognition. They show a lot of with known fungal virulence factors, one way of annotating them. Individually, majority can induce ETI, but in the context of other effectors, some can induce ETS or ETI, but majority don't. This is what they call a meta-effector effect. That means even though the single effector might induce, turn on the defense response, but the other effector can dampen the effect of the immune response effector. So that means the end result, you get disease. So there are only small effectors that can cause ETI, ETI and PTI, but however, the other effectors will dampen the effect of it. So important thing is, so if you can turn on, remember the whole concept of PTI and ETI and implanted immune system is that PTI is a first level of resistance, ETI is a second level of resistance, typically associated with the R genes or resistant genes. We know, Fusarium does not have FHB resistance, not through it's quantitative. It's not through any kind of gene for gene response. So what we're observing here is that what do we see at ETI? What is all about? It might go through our gene response, but doesn't have to infect gene for gene response. I think something novel you got to think of is that it might trigger the whole resistance complex that is independent of the R gene, right? So that could be happening here. But in order to give resistance, you need a combination of both the PTI and ETI to get a robust resistance. So there's something novel that we observe in this. It might analyze the can effectome in this analysis. So in the last few slides, what I'll do is that question of what are the mode of action? So great, we have discovered all these effectors that are doing is turning on immune response and plant. What are they actually targeting? How do we go towards? How do we find out what they're actually going after? So this approach we're taking, I think called turbo ID technique. Some of you are aware of that, most of you might not be. It's a very straightforward technique. Essentially, we take a bait, which is your effector, we tag it to a BRA, which is an enzyme. It's called biotin ligase. What it does is that the biotin ligase will put on a biotin on the protein that's surrounding it. So for example, this little orange blob here, these are like a 10 to 20 nanometer diameter in terms of region where the protein is expressed. So when an effector is expressed along with your BRA, anything protein around it will get biotin labeled. What is important about biotin labeling? As soon as a biotin gets labeled on a protein, it's covalent. It's resistant to many kind of denaturing conditions, which is exciting because you probably know most of you done immunoprecipitations. This requires a lot of washing. That means quite often you lose a lot of the transient proteins. In this case, even to denatured protein, you do stringent wash like SDS or anything kind of washes, it won't go away. It's still there. What you then you do is that get rid of all the biotin, get the SDS. You can pull them down using streptavidin beads. Streptavidin biotin is one of the strongest interactions known, like 10 to the minus 6, incredible affinity. So using that interaction, you can pull them down, then do a mass spec analysis. This is done by our collaborator, Morton Chris Rampage. Without him, many of this cannot be done, obviously. So how do we do this? What we do is, because the question is that you first want to see if there's effective any function in our this kind of assay. So what we do is we take our effector of interest, we inject them into benthymiana using agrobacterium, which is a very common technique. And here's an example of what you see here. For example, here the 9972, you notice that on his own, the red block here on his own doesn't seem to do anything. Okay. However, you have a control here. These are called the BAX and INF1. These are well-known inducers of plant defense and cell death in benthymiana. You notice on their own, in the inclusion of the beta-tubulin, we use that as a control. You can bait on its own, doesn't do anything. You can see that here. Combination of, if you do the BAX as well INF1, they induce cell death in benthymiana. We notice that here, in the presence of the effector, the BAX-induced cell death completely disappears. So what we have in this scenario is this particular effector, they suppress the defense. Therefore, you see the effect of that here. So that's the combination of these two here. On the other hand, but you notice that INF1 doesn't have any impact at all on INF1. That means two different pathways. The cell death path, the INF1 and BAX go through two different pathways of inducing cell death. This is mitochondrial induced, other one is normal cell death process. And here's another example, 4563, where you notice on his own gets induced cell death. That means inducing defense response in benthymiana. But notice in the combination, it actually exaggerated. So both INF1 induced and BAX-induced cell death in the combination effector causes more cell death. So what you have is it's a clear example of individual effectors can either suppress or activate immune response, and even in benthymiana, as I already showed you, the ETS, ETI in wheat. It's a nice corroboration of the result independently in the different pathosystem. So what do we do? So what is the next step? So what do you do to take the in our case, we use this thing. We tagged our B-Ray, our turbo ID, with our effectors. We used to 9 effectors. We used the 44 effectors that we in our core effector. None of them are nuclear coded. That means they enter nuclear, at least mathematically predicted to be enter into nucleus. So I got to tell you the reason why, because also a good way of, because we isolate, because we inject the nuclei, 2 things we can do. Because when you grind a benzimidine, it's like lots and lots of proteins, cytosolic proteins and other proteins. So by looking at the nuclear effectors, what we do well, after injecting, we wait for like 40 to 72 hours. We can actually enrich for nucleus. We can grind the leaf up. We can isolate the nuclei. It's not completely pure, but the important point is we enrich them. We get rid of like 90% of all the garbage proteins that's in the cytosol and other proteins. So we know for sure if you find your effector in there, we know it could they're nuclear targeted. So that's what we said. Okay, let's take 9 nuclear effectors. We inject them. We isolate the nuclei. And then this is what we found. Out of the 9 effectors, we did the mass spec. This cut it. I must admit, this is like a lot of protein work on into this. I'm trivializing how easy it is to do, but it's not. It took a lot of optimization. Each of the effector done like 5 or 6 times into the bioreplicas, over 20, between 15 and 20 control things. So we have a lot of so-called, not necessarily properly interacting. So the control proteins that are non-specific protein. That's the word I was looking for. Non-specific proteins that are used in control can be get rid of. We make a database for that. So anytime we find a protein, we see if it's not there, great. That's our effector targets. So based on that, based on stringent conditions, we identified 170 potential plant targets. They're with the function in terms of annotation, they range for quite a range, all the way from involved in RNA degradation or RNA interaction, lipid modification, secondary metabolism, whole range, gamut of is function. But here is something. Here's much more nutshell in terms of exact what are the class of plant targets. So, for example, we actually discovered some of the previously characteristic immunity, like EDR4, stands for EDR enhanced disease resistance, EDR4, which essentially what chaperones other protein called EDR1, which we know is involved in resistance. What it does is that EDR4 takes EDR1 from the cytosol into nucleus where the function is. So basically it's a EDR acts like a chaperone. So imagine if an effector targets EDR4, it prevents EDR1 coming into the nucleus, screw up its function. So it looks like we have at least, but one of them we have characterized it. We also discovered, for example, we talk about how the effect can target hubs. For example, compass complex, hugely important in as large complex proteins involved in epigenetics of control the epigenetics of chromosome, things like that. So that's also common hubs. We also have common targets. Remember I mentioned the redundancy in the effectors. We're discovering that as well. We found at least 5 or 6 effectors targeting the same, complex of proteins in the nucleus. That includes proteins involved in, PCD, programmed cell death, which is exciting stuff. So we also, there's all signaling pathways. So, quite often, as I mentioned, there is redundancy in the immune system. So you can have a, SA pathway, like multiple genes, SA pathway, signaling pathway. What we're finding is that effector targeting, multiple effector targeting different genes and signaling, proteins and signaling pathway. In this case, I give an example, chromatin remodeling. We found multiple proteins in the chromatin remodeling complex are targeted by effectors. More than that, we're also finding potential novel resistant susceptible proteins that includes involved in protein modification, very important proteins involved in as a susceptible protein because they can change, things like nucleation or ubiquitination, things like that. These are very important proteins on modifying susceptible proteins in the plant immune system. And in addition to that, we're also finding our new LRR kinase. We haven't confirmed it yet, in terms of its function, and that's what we're doing right now. So these are the ongoing experiments. That means first, are these many of the interactions real? So we're using, for example, the split luciferase assay, very simple assay. Essentially, the luciferase themselves, as you know, causes light. and it can be split upon to N terminal of luciferase and luciferase. When they combine, when they come together, they can turn on light. So what we do is that we fuse A, for example, that'll be the effector with the N loop, and we can fuse the plant interactor protein, the B, with the C loop. If the A and B interact, then the N and loop come together, you get light. So that's a good confirmation, physical confirmation of this is their effector protein interacting with their plant protein. Here's an example of 09972 with one of the interactors. I showed you that before in the Benthamiana, how it induces it suppresses cell death. We can actually show that some of the interacting proteins some of the time can interact 9972 as well. There's a very good indication of the physical interacting those things. So right now we also, and the one thing we had to do next step is that are these proteins, plant proteins, involved in the immune system to begin with? So where they have the genetic approach, we're taking many of the plant proteins, we're finding a knockout lines, or we're using the Arabidopsis for that one. But right now we are just in the process of processing some of these seeds, as well as ordering more than knockout. We're working about between 30 and 40 construct plants right now, knockout lines. So it's going to take a lot of work. But anyway, it's ongoing. We'll get an idea of what's going on there. All right. So the last 2 minutes, I'll finish off my webinar. So yeah, with respect to the fan effectome study, we can identify core effectors. One of the cool thing about core effectors is that I mentioned either core effectors found within the species complex. What we're finding, especially using the AlphaFold 3, is some of those effectors at structure level and actually have homology or structural homology to other plant pathogens, completely unknown pathogen involved in disease in wheat. I'm collaborating with Hossein Borham at Saskatoon. We found like 3 or 4 effectors in our core proteins that are structurally homologous to Leptospira maculans, which is fantastic. That means what you think about is that is it possible that these effectors might be targeting some common host protein, and that could be a potential example of a target for manipulation, right? If you have common protein that's been targeted with effector for multiple pathogens, if you mitigate that, you make it provide resistance to multiple pathogens against multiple pathogens. That's a cool way of looking at it. I'll give you the example here, just to emphasize the point of common proteins. Here's an example: WA1, which is a wheat-associated immune complex, a ZAR1. So if you don't know what the effector is that targets this protein, resistant protein, here's a ZAR1. Again, Arabidopsis has targeted by a bacterial effector, and here's SR35 targeted by a stem rust in the wheat. So what you have, you notice that is that at the structure level, especially SR35 and ZAR1, they literally overlapped each other. So you have a common R gene that's targeted by a stem rust and a bacterial effector. So this is very cool. So that means I think we're going to find more and more of these things down the line if we start analyzing at the level of structure level and also the assay we had done before. So by knowing the core effectors, we also can compare the pan effectome to the effectome of new isolates. For example, if we find a new isolate that's probably causing disease, and the question, what do we do about it? So we can just look at the effector compendium in the new isolate and see, and the core effector or even the accessory one, and compare to the pan effectome and see if there anything changing happening. We've been able to identify which effector or core effector has been modified or ongoing changes, and that's what we can go after as a good surveillance tool, right? We can monitor them, and then we can also, once we know what they are, we can actually manipulate them. We can actually go after them and target the core accessories. So using mitigation strategy, like people can use them peptide binders or even SIGs, the spray-induced gene silencing. The various techniques out there, we can start targeting effectors. In fact, our collaboration recently with Steve Robinson in Saskatoon, and some of the targets we provided to him, he's able to show one of them at least has a dramatic impact on virulence in FHB in the field studies. So by just using this, understanding the core effectome, and then this at the structural level and important how they play in disease with all that assay I've shown you, we can use a very directional approach, very direct approach. If we're not just targeting any gene of interest, but really targeted approach to go after core effectors that are important for disease and able to mitigate them. And the last but not least, I mentioned to you by understanding some of these plant targets, including the so-called plant common resistosome, able to pyramid resistance proteins. That's our normal thing people do these days. You can pyramid on different R genes, so they're able to provide enhanced resistance, not necessarily to Fusarium, but also to other pathogens as well. And you can also using, for example, by using techniques like peptide binders, you can actually target the S protein. By targeting them, you can also induce immune response. Provide that could be by using peptide binders. Are used. It's not transgenic, but it's very temporary. So you provide very immediate activation of our genes by not, but by the same time, not screw up the function of the S protein that may be involved in, for example, plant development, for example. So we don't want to screw that up. And finally, we can also activate our proteins. Instead of pyramiding the resistant proteins at genetic level, we can artificially activate our proteins using what we call them therapeutics. We have, for example, in our lab, we're doing chemical screen. Previously, we identified things like Zeractin, which is a small molecule. Was actually able to activate the ZAR protein, independent, and just a combination of ZAR and Zeractin and the effector, able to activate the ZAR protein and able to enhance resistance to, again, in Arabidopsis against other pathogens. So this is fantastic. So you can actually turn on the immune response without genetic approach by using chemical biology to turn on the R genes. Again, it's also transient. So you don't have to worry about turning on like a constitutive turning on the R genes and decrease the yield for that matter. All right, so I'll stop that. You got 10 minutes left for some questions. And here are the people I have to acknowledge, obviously. Chris Blackman, I mentioned, pretty much 99% of the work that's done here, that's here. And Tian Lee, he did all the structural stuff here, that's him. And Malik also contributed some of the knockout lines that we have shown in Fusarium, and that's here. Margaret, that's our technician. So if you thought her, we won't run the lab. And Alexia, unfortunately, I can get a photograph here. She also contributed quite a bit to the all the knockout studies we're doing in the lab. Also the Chris Apple's lab, and also for the mass spec studies. And Daryl DeVoe, my longtime collaborator in UFT, we worked together for over 25, 30 years and looking at the plant effectors and plant response immune system. So I'll